Hi, I'm Laura and I will be explaining the chemistry behind chemotherapy. Chemotherapy is one of the most used ways to treat patients with cancer. So before we get into the chemistry of it all, let's start from the basics. What is cancer? Our body has trillions of cells and cancer develops in a cell. Each cell has genes that act as an instruction manual. This instruction manual mainly tells the cell how to grow, work, divide, and die. This is known as the cell cycle. There are many parts of the cell cycle, however, this video will focus on mitosis as that's what cancer is from, since that's where cells replicate. There are checkpoints throughout the various states of mitosis that keep the number of cells that are being duplicated and destroyed at a controlled balance. However, there are times where a mutation happens and a cell will not recognize these checkpoints. Therefore, it will not stop and will start growing cells uncontrollably. These cells are abnormal and can create a lump known as a tumor. This tumor can either be benign, that stays in one place and is non-life-threatening, or on the other hand, it can also be a malignant tumor, or it can spread through the body and become life-threatening. Chemotherapy is a drug treatment that uses strong and powerful chemicals to kill these fast-growing cells and stop them from spreading. Chemotherapy drugs target fast-growing cells at various phases of their cycle, and since cancerous cells grow much faster than normal cells, it makes them an ideal target for these drugs. However, chemotherapy drugs cannot distinguish between healthy and cancerous cells. Therefore, these drugs can also damage healthy normal cells, which is what causes several side effects. However, the normal cells that have been damaged have the potential to recover once chemo is over, unlike cancer cells. This causes side effects such as fatigue, hair loss, easy bruising and bleeding, infection, and many more. There are many chemo drugs that can be used, and it is very important for doctors to know how each of them work and their chemical structure to minimize side effects and know which drugs will go best together, as sometimes more than one chemical is needed for each different patient. Doctors are also able to determine the dose and which drugs to use depending on the timing of the cell phases. These types of drugs are alkylating agents, nitrosamines, antimetabolites, anti-tumor antibiotics, topoisomerase inhibitors, meiotic inhibitors, and corticosteroids. However, this video will focus on three main chemo drugs that interfere with the DNA of the patient, which are alkylating agents, nitrosamines, and antimetabolites. Alkylating agents is a chemical that will prevent the cells from replicating itself by damaging its DNA. What this means in the chemical world is that they need to be able to bind to phosphate groups, amino groups, sulfhydroxyl groups, hydroxyl groups, and imidazoles groups. This drug is used to treat cancers such as lung, breast, ovarian, leukemia, lymphoma, myeloma, and sarcoma cancers. The risk of these drugs is that because it damages the DNA, it can damage the bone marrow where blood cells are formed, which could lead to leukemia. However, this depends on the dosage the patient is given. Some examples are meclorothiamine and cyclosporamide. Nitrosamines are also a type of alkylating agents that have a special role. They inhibit DNA repair and are able to travel to the brain by crossing the blood-brain barrier and treat brain cancers, unlike other alkylating agents. The genetic structure of a nitrosamine are hydrogen atoms found in the R1, R2, and R3 groups. Their structure is able to degrade carbonium ions, which is an electrophile, which means electron pair acceptor, and damage guanine, cytosine, and adenine, which are nitrogenous bases that are found in DNA. Some examples are carmustine, lomustine, and streptozotocin. Antimetabolites also interact with DNA and RNA by being a substitute for the building block of DNA, which is a phosphate group, sugar group, and one of the nitrogenous bases, adenine, guanine, cytosine, thymine, and uracil. This disrupts nucleic acid synthesis, which is the process of synthesizing or degrading DNA and RNA. The reason why they are able to bind to the building blocks and act as a substitute is because their chemical structure is very similar to the molecules the body uses. There are two main types of antimetabolites. There is purine analogs and pyrimidine analogs. Purine refers to the nitrogenous bases in DNA, which are adenine and guanine, and pyrimidines refers to cytosine, thymine, and uracil. It is less clear how the purine analogs work. However, pyrimidine analog contain fluoride atom 5 carbon positions on the ring. And an example of a pyrimidine analog would be fluorouracil, and an example for a purine analog would be thioguanine. So that is the main overview on the chemical side of some chemotherapy drugs, and I hope you learned something. Thank you for watching.